Okay, hi everyone. So a lot of the work in my group is looking at trying to connect pure and applied ecology to understand the role of environmental stresses, how they operate, and how they affect complex systems, multi-species systems. So here we're focusing particularly on food webs and other types of ecological networks. So this is just a highlight here, some of the work that the group has worked on. We've looked at pollution, effects of flooding, drought, land use change, and also warming. Today I'm really going to focus on warming, drought, and a little bit of our pollution work, and just give you a broad overview of some of the work we've been doing. So this is the general sort of framework within which we operate. We're trying to connect different levels of organization to understand how these complex multi-species systems operate, but also how the lower levels of organization, from the sort of population down even to the genetic level, can influence the higher levels. So when we're dealing with complex systems, one of the first things we need to try and do is to see if there are any kind of either recurrent patterns or perhaps simple rules that we can use to try and understand them. Simply because a system is complex doesn't necessarily mean it's unpredictable. The cell cycle is extremely complex, yet it is also very well understood and predictable. So one place to start, which has always seemed fairly obvious to me, is that in aquatic food webs in particular, which is where much of our work is focused, that there is a strong structuring role of body size. Body size and abundance are two really key things that strike you when you look at any aquatic ecosystem. And this led to our formulation in a paper in 2005 in Tree, which followed on from Joel Cohen's work in PNAS on Tuesday Lake a couple of years previously, where we collated data where we had mass and abundance information for food webs. At the time, there were only these three that we could find. Broadstone Stream, which I had worked on, Tuesday Lake, which Joel Cohen and Stephen Carpenter and others had worked on, and the Eithne Estuary, investigated by Dave Raffaelli, etc., in Scotland. And we did find that, sure enough, body size and abundance had clear roles in the food web. Here, the nodes represent species populations, and the lines between them, trophic interactions. And all we have done is simply plotted abundance here on the x-axis versus body mass on the y. So what we can see is that there is a general pattern of energy flux moving up through the web into the ever larger organisms from from small and abundant organisms to larger and rarer organisms. And in fact, these three webs all fit in pretty much the same sort of phase space if you draw a common regression line through them. So that gave us some hope that we could actually use the food web as some kind of ecological prism to try and understand what's happening in response to stresses, but also by linking different organizational levels together. It kind of forms the nexus really between community and ecosystem ecology. And this is really the framework within which we're operating as a whole. So we have our extrinsic drivers here, which could be nutrients, or pH, or climate change. This has effects on biodiversity, species traits, abundance, and interactions, and also the individuals that carry out those interactions. These combine to form the food web, which has attributes such as web size, the number of links, the strength of links, as we've seen in the previous talks, which are important for stability, which affect ecosystem processes such as decomposition, biomass production, and in turn ecosystem goods and services, which are important for humans. So I'll just give you a couple of case studies to show some of the work that we've done applying this sort of general framework. Firstly, I'll look at some of my older work on acidification and recovery from acidification, and then more recent work exploring climate change. So acidification, it caused widespread species losses and collapse of ecological networks in the 1970s, particularly in Europe and North America, and in fact is a growing threat now in emerging economies, particularly in areas in India and China. So it is a problem that has not actually just gone away, although it is often thought of as being a 20th century problem. So we have seen widespread collapse of fisheries from the top of the food web being eroded down, and there is a general relationship between pH on the x-axis here and the size of the food web. This is a log scale of the macrofauna in the web across over 100 different ecosystems in the UK, the standing and running waters. As the system becomes acidified, the food web simplifies. We also see not just structural change in the network, but also changes in its stability. We carried out some research where we constructed food webs from 20 systems, and we had mass abundance data. We could characterize and infer interaction strengths. We then modeled the robustness of these systems using dynamical simulations. And one thing that we found was that in these more acidified systems, they tended to be more dynamically stable. And this sort of tied in with some suggestions that we had that, in fact, although we've seen quite rapid chemical recovery since the 1970s as acidifying emissions have been reduced, the biological recovery has not kept pace with it. It's tended to be rather slower. And there are suggestions that there's potential ecological inertia within the food web which slows the biological rate of recovery. We do see, for instance, some interesting patterns when we look at the food webs which maybe seem a bit counterintuitive from classical biomonitoring work. Here, for instance, we have data from the 1970s, 80s, 90s, and 2000s in Broadstone Stream Food Web, one of the model systems our group has worked on, where again we have mass and abundance. Now we have switched the axes. This is more statistically correct, although perhaps maybe not necessarily so intuitive. So here we have larger things on the bottom in this end and smaller things at this end. What we can see here is in these food webs, which are sort of enclosed in these bubbles, from the 70s to the 80s to the 90s to the 2000s, what we see is pH is rising, there is chemical recovery, but invertebrate abundance is declining. Now, if you just carried out biomonitoring looking at invertebrates, which is usually the case, that would seem to make little sense. The reason for this is that over time we have increasing abundances of larger predators arriving in waves. Here in the 70s, this is the head capsule of the largest predator in the system, which you can probably barely see. This is the one that dominated in the 80s, in the 90s, and then in 2000s we have this chap arrive, the brown trout, which is this red blob here. As these ever larger predators come in, they exert stronger top-down control on the web, and it suppresses abundance and skews the structure of the food web. So understanding network structure and dynamics is important for understanding ecological recovery. So that's some of the work that we did in a nutshell, really, on acidification. More recent work, we've looked at climate change. And in particular, we use a range of approaches combining surveys, natural experiments, and field experiments. None of these are perfect. We have to use a combination to find the answer to the questions we are posing. So climate change itself, we need to bear in mind, is a compound stressor. So there are potential synergies that can arise between its component parts. But before we can understand that and climate change as a whole, we need to try and isolate these different stressors and look at them in turn. And I'm just going to give some examples in the talk today about hydrological change via habitat fragmentation and habitat loss due to drought, and temperature change, which can cause elevated metabolic demands at the individual level, which filters through to the ecosystem and food web. And much of our work is increasingly using mesocosms to study climate change. There is a review paper led by one of my students recently which considers this sort of growing field and evaluates it. I'll give two examples today of where we have used pond mesocosms to investigate warming and stream mesocosms to investigate the effects of drought. So drought first. These are results from a long-term experiment which ran for a couple of years where we imposed drought treatments on stream mesocosms. Here is the drought treatment, which was dewatered once a month for 6 days. It wasn't completely dried, but most of the water was drained out and then re-wetted, versus a control treatment which was continuously wetted throughout the whole period. And these data here are just showing replicate food webs with, again, mass abundance scaling for the control treatments. What you can see here is these all look extremely similar. So we can actually produce quite realistic and replicated food webs in these mesocosm experiments, which gives us some confidence that we can generalize to real systems. One thing that we find is that drought simplifies the food webs. Here we have the reference or the control system versus the drought system, you can see that many species have disappeared, particularly from the higher trophic levels. Larger things disappear, as do rarer things, and also ecosystem functioning is impaired. Secondary production, shown here by the size of this bar, declines in the system. So total system productivity declines by about 1 order of magnitude, and the food web simplifies. It loses about 1/3 of its species. So we see quite dramatic impacts of drought in these food webs. Well, what about warming as a different component of climate change? This is where much of our work is being directed now. As we've seen in previous talks, we know that global warming projections for the 21st century are predicting rapid rates, particularly in the Arctic. So this is an area where much of our work is done, a long way from severe, but it's a good model system for studying particularly rapid rates of change. And this is where we've been working recently. Now, temperature is a key biological driver. And we know that body size drives metabolism. It is from diatoms to blue whales. It shapes the metabolic rate of an individual. We can see here mass and metabolism scaling from single unicells through to the blue whale. So a highly predictable relationship. We also know that body size affects food web structure and dynamics, as we've seen from our mass-abundant scaling relationships. But importantly, metabolism is driven by body size as well as temperature. It's these two key variables that we're interested in. They affect the individual, but they can also scale up to the whole ecosystem. Now, this graph here shows the typical approach of much of food web ecology to considering populations of individuals all being treated as being identical, as we see here from our Liverpool fans. In reality, of course, there is a huge amount of individual variation that filters through to these different levels of organisation and helps us to understand what can happen at the ecosystem level. So we'll just have a look at some examples of these different organisational levels from studies that our group has been involved with. This is data from a mesocosm study where we had a set of 20 ponds warmed to different degrees. So blue is ambient, and then each one had a paired pond which was 4 degrees above ambient, and these have been warmed since 2006, so again another long-term mesocosm study. What we see here is that the average individual body size of an organism within the phytoplankton decreases by about an order of magnitude, whereas in the zooplankton it stays fairly similar. These changes associated with warming are generally quite widely predicted from Dufresne et al. And various other studies, and are closely linked to predictions associated with the metabolic theory of ecology. So here we have data that seems to match up quite nicely with what we might expect from theory. But these mesocosm approaches, they are all very well and good, but what we really need to do is to try and scale up, to move from these sort of patch scale approaches into full ecosystems, and then to consider particularly the wider context of sort of meta networks in the landscape. And we see a bit of a disconnect really in many ways between some of the sort of food web models that are widely used and where the data are actually collected. So we're trying to bridge the gap between these two. One way in which we do this is by working in these Arctic and high latitude systems I touched on earlier. So here we have Iceland, which is just on the edge of the Arctic Circle. These are very useful as they act as sentinels of climate change. They're sensitive to temperature change, and we are predicting, or they are predicted to have the fastest rates of change in the near future. So this is a map here of the system that we work on. This is a catchment in the Hengil region of Iceland. It is a geothermal catchment, so these streams are warmed by geothermal activity, but importantly, they have very similar physicochemistry. The one thing they differ in is temperature. They range from about 5 to 25 degrees Celsius in this thermal gradient, and they are all within a 2 kilometre range. So we don't have dispersal constraints. We have a temperature gradient in a natural system, and importantly, we don't have to pay for the heating bill. It's all free. This is just an example here of the nice paired nature of the design here in its most perfect setting. We have streams that are 2 meters apart, but separated by about 15 degrees C in temperature, and they have very different communities, very different ecosystem processes. So I'll quickly run through some examples of the different levels of organisation that we've been looking at in these systems as we move from the individual through to the ecosystem, and give examples of the effects of temperature. Well, firstly, one thing that we have noticed in recent studies which have been published this year and last year is that, in fact, there is little compelling evidence for this third universal rule of warming, that things get smaller as they get warmer. In fact, what we've found is that in all the species, 67 species we've measured, body size decreases in 8 and increases in about 7. So clearly something is happening in these systems which isn't really in line with the typical broad brush theoretical predictions. At the population level, we see strong impacts of temperature. We see abundance of certain species, such as these filter feeders, increasing, and these algal grazers, these snails, increasing especially rapidly after some threshold level. So there is a strong effect of temperature on population abundance. These species benefit enormously, other species do not. Here we have a rapid decline over the temperature gradient in a chironomid species, and this one here has a kind of threshold level and then it collapses. So all of these population changes occurring within the community, of course, ultimately lead to community level shifts. What we have in this graph here is the pairwise temperature difference between all the different streams in the system for 2 years, in 2004 and 2008, and a measure of community similarity on the y-axis. So 0 is no species in common between a pair of food webs, and 1 is all species shared in a pair of food webs. And what we see is that in both years there is a general decline. As temperature difference increases, so does community difference. And this just highlights here for these two streams, very close to one another, physically identical. They only share about half of their food web nodes together. Within the structure of the food web, we also see changes in food chain length. So it is not just the nodes that change, but also the connections running through the food web. Here we have a measure of food chain length using delta 15N, a stable isotope measure. What we see is that as temperature increases, so does the trophic height of the web. In fact, over this temperature range, we essentially add an additional trophic level to the web as fish enter the system. So again, this is perhaps a little bit unusual that we get large predators being added as the system warms up. And one thing that we see in particular is the very tight coupling, this keystone interaction. When the snails dominate in the system, as temperatures increase, we also see the increase in the fish. So what we suspect is that this is a kind of keystone interaction. Because these are extremely efficient grazers of algae, the energy from carbon fixation gets into the system and then through to the fish very rapidly. So we have this general scheme where larger predators with longer food chains are supported by these grazers. There is intense pressure here, but the system can withstand intense pressure because there is very high primary productivity and very rapid nutrient cycling. So this shows you these interactions in the context of the wider food web. Here we have the trout in the warm systems at the top of the web. Invertebrate predators are largely missing. We get a few, but not many. We have these primary consumers here, but the trout, in fact, are also cannibalistic, so they have an extra trophic level within the same species. And then down here, a large array of diatoms, the unicells. In the coal system, we have a similar number of species, but completely different identities in many cases at the base of the web. We have lost the higher trophic level, the trout are now missing, and we have invertebrate predators coming in. So very different network structure, different food chain length, and potentially different stability in these systems. This is an example here now of some of these trivariate food webs with mass and abundance, as we've seen in the previous studies, highlighted for a cold stream of 5 degrees versus one of 20 degrees. What we can see is, particularly when we have the trout in the system, we have this potential effect of the larger, more abundant predators
possibly being there due to increased resources, potentially more efficient energy transfer. But because these are very large, there is the potential that they could have destabilizing impacts on the network, which is something we're investigating now. So these mass abundant scaling relationships, they're kind of underway. We have done about half of the streams, so this is very much work in progress. I'll have to report back to you on this later. But one pattern, an intriguing pattern that is emerging, is that the complexity of the system declines as temperature increases. It is possible, and we will carry out modelling work to investigate this, that there is an interplay between complexity and stability. The systems might be more simple, and this could modulate network stability. But I'm wary because we only have half the data, so these relationships might not hold next time, but hopefully they will. We will see. So these are impacts on the community level, the food web level. We'll have a quick look now at warming impacts on ecosystem functioning. And what we see is, perhaps unsurprisingly, many process rates are strongly related to temperature. As temperature increases, so does decomposition by microbes and also by detritivores, and also algal production. So both inputs to the food web, algal and detrital, increase. Nutrient cycling also speeds up. Here we have NH4, NO3 and phosphate, so nitrogen, species and phosphate, different nutrients. In cold systems versus warm systems, we can see that the cycling rate increases dramatically, but to differing degrees. And these ecosystem process rates, what we find is when we actually carry out translocation experiments, is that they are essentially identical. We can take very different communities, and the temperature response for ecosystem respiration is exactly the same. This was carried out from 4 different streams of very different temperatures, and they all fall on a single common line. And importantly, we see congruent relationships between ecosystem respiration across all of our Icelandic systems to what we see in our experimental ponds in the UK, which gives us some hope that we're dealing with potentially universal patterns and responses. And in fact, in more recent work, which one of my students published last year in Nature, we also see congruent patterns across hundreds of different aquatic and terrestrial ecosystems, suggesting that we have a good model system to play with. More recent work now is we're trying to address these potential transient versus equilibrial dynamics because the streams have been warmed for a long time, potentially. What happens when we impose a short-term stress on them and within intergenerational time? We now have an experiment where we are manipulating the temperature of this cold stream by warming its water, again by about 4 degrees or so, and 1 year of warming has just been done. And what we see already, although I don't have data to show you, is that there is a rapid response. Its community now resembles the warm streamed. So there are some unanswered questions that we're addressing with ongoing research now. We are asking, well, why are top predators so abundant in warm streams, carrying out predator manipulations? And we think that perhaps terrestrial aquatic linkages, which we're investigating now, could be important. And also we're interested in microbial metagenomics and metatranscriptomics, trying to understand how the molecular level of change could affect ecosystem processes. And to do this, we're also trying to expand our viewpoint, moving from Iceland into other systems. So Svalbard is one system in the high Arctic that we are now looking to replicate this work and to take it to the next level. And in fact, this summer we have identified geothermal systems or naturally warm systems in Kamchatka, Svalbard, Iceland, Greenland and Alaska. So this is where we are going in the future. The idea is to try and connect from laboratory work and models to mesocosmostyle studies to a more global perspective, ultimately. And I'll just finish with some nice photos of some of the field work that we've been doing recently. This is Svalbard. There are quite large logistic challenges. We can only get into it by yacht. In Kamchatka, we can only get into it via Russian helicopters, which is even more dangerous. But what we can find is when we get there, we can replicate our model systems. This is Kamchatka. It looks almost identical, although vastly different in species richness, to the physical environment in Iceland. So we can find the same systems to work on in the future. We do, however, have some additional concerns with larger predators being added. Brown trout is the top predator in Iceland. Here we have something much more dangerous. And in Alaska, you find that they're not the only top predators you need to worry about. This is a sign outside one of our field sites telling you that they are insured by Smith & Wesson. So enter at your own risk. And in Greenland, of course, it is even more dangerous. So the field work is quite exciting, really, and my students have to fill out very detailed risk assessments before they're allowed out of the classroom. And just to finish, so this is the work that we've been doing in these exotic, far-flung places in the north. A bit closer to home, we are also applying the same approaches. Mike Bruford touched on this, a large-scale project being led by Cardiff University called Duress, where we are looking at food web responses to climate change and land use change across Wales, using up to 400 sites and 20 intensively constructed food webs. So we are slowly being able to sort of generalise and expand the Icelandic work to other systems at high latitudes and the temperate zone. So thank you for listening.